So hi, I can see myself online, so I suppose you can also see me. Hi, hello, and welcome again. I'm Oliver, Microbehunter here, and welcome to another live stream, a microscopy live stream. Yeah, a little bit today had a little bit of a different time. Normally I do the live streams on Saturday, a little bit later. Um, and I can also hear myself now. Aha, very good. Okay, yeah. So I think we're all start. Yeah, yeah. And last week I did not do a live stream because I was not around. I was for several days away. Also could not prepare a video, and yesterday I was also busy, so I decided to move it to today. I'm glad that you could still join me. Hope that yeah, the time is more or less also suitable for you. Yeah, so what I would want to do today is the following. You see over here some paper-covered microscope slides. Those over here, those red ones are antique slides, maybe from the Victorian time. And a couple of years ago, I decided to experiment a little bit by paper covering my own slides. So these are here already a few years old. I decided, well, why not pick up on that again and try it again? It's honestly, I think it's pretty useless, almost, because why would you do that, right? Yeah, they do make the slides a little bit more stable. Yeah, sure, maybe they look nice for branding purposes, maybe as well. It's more kind of keeping up a nice old tradition of covering paper covering your slides. And if you're interested a little bit more, I did find a couple of online resources as well. I already put them into the description. There's a really nice online article about making paper covered slides with a couple of other references. So please do check that out as well. So what I'm gonna do therefore today is I prepared yeah, so yeah, a little bit of paper here. If you want to have those templates as well, there's a PDF and also a PowerPoint file in the yeah, also in the description that are linked if you kind of also want to try it out. Yeah? Again, this is a little bit semi-improvised as always. Yeah, what I'm going to do now is I'm just going to go through again a little bit here. Yeah, hello from Florida, from England, from India, from the Philippines, Germany, Philippines, France, Ukraine, and from Netherlands. Okay, and quickly check here as well. Just a second, some organizational stuff here as well. Yeah, 31 people have already joined in. So apparently this yeah, this time and day is at least for those of you who are here, it still seems to be fine. Yeah, so that's basically one of the things, and I do also want to show you a couple of other things that I've been working on over the last couple of days. But I would like to simply share with you a little bit of what I've done here. And I'm going to, but before I do that, I want to show you those here a little bit. Again, these are fairly antique slides. See, this one here is already broken a little bit. Inside here, the slides are also thicker. This is how it looks like from the other side. Yeah, yeah, you can see that they do have a fairly nice pattern. Yeah, also a space to add a label, and yeah, some slides from I don't know 100 years, 150 years ago. They were actually quite nicely paper covered, and companies and slide makers used them to actually brand yeah, their slides. If you want, we can have a look at them as well. Needless to say, those paper covered slides, yeah, you got to be careful. Do not use oil immersion, obviously. Because otherwise, yeah, it's not going to be good for the paper, right? But I think it's a nice tradition that we can try to keep up, and I'm going to put those away here because I'll be working with a little bit of glue. Yeah, what I've done here a little bit is a couple of years ago I experimented around making those slides here. I also tried to add some clear, how do you call this, clear paint over it, also to make it a little bit more waterproof. So I've been experimenting around, and yeah, yeah, they are a little bit more stable. But I would say that, yeah, maybe, yeah, you probably would like to think twice of actually doing those. So actually, only those that you really value probably kind of pays off. So you've seen that I've also experimented a little bit by printing it directly on the, yeah, on this paper over here, adding all the separate slide labels and so on and so on. Yeah, and for a video that I made several years ago, I even added this label over here. Okay, yeah. So what I want to show you is I want to show you first a little bit of what I've been doing. and how I've been, so we just stack them here like this. And the first thing that I want to show you is I printed out, and if you want, you can download it. This is basically what I made using PowerPoint, right? And so there's a PDF and also a PowerPoint presentation that you can have a look at. And it's basically, I've made those drawings here. And the idea is I've got a couple of slides here, and I just want to paper cover those and to just show you how I've done that, and then just take it from there. Of course, everything over here is, this is actually the wrong one because this one is a little too large, but this one over here should be right, and you see that it fits directly in here, right? And then if you see those double lines over here, that's basically the edge, 1 millimeter of the edge of the side of the slide. Then you kind of fold it around over, and then you add another kind of paper on top like this. You glue it on, and then you can always add some kind of a slide label later on as well, right? So that's kind of a little bit the idea. I just see that maybe the slide label over here is a little bit too big. Yeah, for my taste. Yeah, but I mean, you get the idea. That's kind of the principle a little bit. And what I would like to do now is I would like to show you a little bit of what I've done in the past. A couple of years ago, there is also a video that I made already before. So yeah. So basically that you can also see it. Be sure to calibrate the printer. That is that's the magic scissors one. Yes, yeah. It's a little bit of an inside joke. I did make a video, where basically I was cutting out those with scissors. So, but I tell you the most difficult part, and maybe one of the things that I would like to change for the future is how do you actually get those holes here, right? And I used a hole punch like this. And then with a hammer. However, what I found out is that the success depends a little bit on how hard or soft the surface is. If the surface is too soft, then it's going to wrinkle and warp it quite a bit. So you want to use some kind of maybe some cardboard on the bottom here, and then it works reasonably well. But there are also hole punchers that can be used. So maybe this is not the best solution here, but there are those other large hole punchers that you might use. Yeah? So, and what I would like to do now is I'm going to do the following. I'm going to use a piece of paper here, and I'm just going to do it like this here, and we're just going to try the first one. Again, I've done this the last time that I've done this was quite a few years ago, and I'm going to do it like this. And what you do is you add a little bit of white glue on here, if I can get it out. Here we go. Okay, here we go. Okay, you add a little bit of glue, and then you take some cardboard, like the one that I have over here, and you just do the following. And you want to keep those. You want to keep those little things here, right? And you want to put one of them in here. Okay, simply to prevent the glue from actually reaching the other side here. And then you kind of want to spread it like this. Okay, and that's a little bit the You want to kind of spread it like this. Yeah, maybe not even enough. This is a water-based glue. Okay, so, and I'm going to just leave it as it is, and then you carefully remove this. Okay, you of course also want to remove the center part here again. Just push it out. Yeah. And of course you want to put it on a nice clean surface over here. This glue dries fairly quickly, and then you want to place this one over here directly here in the center, and you carefully press down, kind of shift it around. Okay, and then all you do is you fold up, of course, the edges here. Okay, you might want to pull a little bit, right? Here as well. I just realized that maybe this here is a little bit too large. So, okay. So that's a Paul nail polish. I have already prepared a label here. So, Paul nail polish. So, and over here as well. And you see that here the bottom is fairly flat. Okay, you want to take care that there is of the glue that might have spread out. You just wait for it to dry, and then you can carefully scratch it off. So that's going to be easily possible. Yeah, and the first slide actually is 50% finished. Yeah. So the next thing that I'm going to do now is I'm going to just prepare this one over here in a similar way. Just put it over here. I also tried to use a glue stick. However, I found out that a glue stick doesn't quite work as well. It does not stick as well. So you have to use plenty of glue here. And the same story again. I'm going to try it like this without actually adding the cutout back. Let's see how well this actually works. Okay. And here again. Okay, maybe this actually also works. So we carefully. You see, that's a little bit the problem that we have. See, there is some glue. Maybe I had some glue over here in the first place, but doesn't matter. I'm going to leave it as it is because as the glue dries, we're not able to see this anymore. And then we carefully place it over here. And I just realized a mistake that I made. Do you actually see over here that I see the black line, right? This was actually the cutout line. I probably should have used the other side. And if it's a little bit wrinkly, then that's also fine, because as the glue dries, it will start to stretch again. So this was the one the one with the pollen in nail polish, right? So what I'm going to do now is I'm going to put this slide label on top. Okay. So, and then of course, if you want, we can have a look at those slides as well under the microscope, of course. You want to spread this a little bit. Again, I'm a little bit improvising again right now. You want to probably take your time a little bit more. This is a little bit more of a proof of concept, and it's not even straight, I just realized. Okay. Yeah, and then one could say we're kind of finished. It will actually look better once everything's dry, because then it will actually stretch and it will look much nicer. Okay. So this is a little bit the very quick, and easy way. Okay. What I'm going to do now is I'm going to occasionally again interrupt myself to read some of the comments that you have written. Okay. Use a scrap of leather under the punch. That's what leather makers use, and it works very well. Okay, thank you. What I used is, as a matter of fact, I then used the same cardboard over here on the bottom side, and then that also worked. I also used a little bit of soft plywood, and it didn't work. So because then it really started to become very wrinkly. I don't know if I have any. Look, here it is. Yeah, I just show you. This was my first trial. If the bottom is too soft, right, then maybe you can actually see the wrinkles over here, right? It will start to bend down, and it did not cut it out nicely. Yeah? So this was a little bit my first trial, and so the bottom line is the lower surface has to have the right hardness, so to say. If it's too hard, then of course you're damaging of course the tool as well. But if it's too soft, then it's not going to cut either, and it's just going to bend here, and you do not get a very nice cut. And what I also found out is, look, that sometimes it will simply not punch through, because I realized that one edge over here is a little bit more dull than the other one. So what I've done is I actually punched it, and then I rotated it a little bit and punched again, and rotated and punched again, to make sure that those hard, the ones that are still sharp, essentially go all the way around. So this was a little bit of trial and error, right? But let me quickly read some of the questions again. Okay. You can get spray-on PVA glue. Okay, that's something I did not try yet. Then there's an off-topic question: what main factors affect the microscope resolution? If I want to get the best resolution without using a technique that relies on backlight, what kind of microscope is best? Well, the very short answer to this: the resolution depends on the numerical aperture. So that's the number that's printed on the objective. I don't have one right now that I do. I can show you later. There's a number print, and that essentially determines the resolution, but there are also other factors as well, right? But actually numerical aperture, that is one of the main factors. what kind of is, yeah, that's a complicated question, like Ever Smaller is saying. Yeah. So integrated circuits on silicon, non-transparent, hence no backlighting. If you want to use integrated circuits, then a non-transparent, then you have to use a stereo microscope. And in this case, when you're using a stereo microscope, you're not exceeding the resolution limit anyway. Okay. So try hard on top and bottom with a template above, so you know where to cut. When doing woodwork, we sandwich on both sides and not to splinter the edge. Ah, I get it. Okay, yeah. So, oops, I placed it on here on this slide. We can try another one actually. Okay, yeah, usually it does not look so nice if there's some glue over here on the other side, but usually if you just leave it and don't wipe it, then essentially it will simply dry and it will become transparent. Okay, so let me quickly do the following. Every now and then I just want to know now. Let me quickly check. There are 35 people now online, so okay, there are actually some people who were able to join it this time. So let's try another one, and then later on I'm just gonna have a quick look at this if you're interested, and I'm going to also show you a couple of other things that I've been up to in the last couple of days. Again, this is a little bit of a, how do you say? Yeah, trial and error. Let's make this a little bit higher. Okay, so let's move this a little bit out of the way. And yeah, again, I highly recommend that you actually visit the link that this is a web page that I found about paper covering those microscope slides with very colorful paper, very nice looking designs. So essentially, this is something that I recommend. You know what I'm going to do now again is I'm going to put again this little piece of round cutout in here just to play it safe. Okay, and yeah, so that's the first one. We lift it off, we remove the center part again, and okay, and the next one, I don't know, that's the onion skin. Let's try this one over here, right? I made this already, I don't know, over 1.5 years ago, so these are not the most valuable slides that I have here, but I did check that the specimen is more or less in the center. So yeah, and again, let's put this on the side here. Let's fold it up. So it's more like keeping an old, more or less old tradition going. I actually also read something interesting today. I found an old a book about making microscope slides, and I read there that they used to even make microscope slides out of wood. So basically, back in the day, 1800 something, they used to make wood microscope slides with a little bit of a glass window, something also that we don't see anymore. So here we go. Yeah, you always have to start on the bottom side, because the bottom side has to be completely flat, because once you fold it over, it's not completely flat anymore, but at the bottom, because that's how it rests on the microscope. So that's number one, and number two. Again, we're gonna try this one here. Now I want to get the correct side, so let's use this here, because the one with the lines should be now the one with the glue. So let's have a look. So we just smeared. Yeah, and in the past I also tried to make my own slide boxes. Yes, I have a how do you call it, a laser cutter, but it's also possible to make slide boxes using corrugated cardboard. So, and that's also something that I might want to show you. Again, all of these things, of course, can be bought as well, but there is a certain thing, it's a little bit fun sometimes to make your own things as well. So, and here we already see a little mistake, and it's also something that you might want to adapt a little bit. Notice that over here, where I was folding it over, that it's a little bit too large, and so it goes a little bit into the round window. So that's also maybe something that I should cut out, yeah, before folding it over. So this is the onion. Okay, and this one is over here. So let's maybe also remove any excess glue and distribute the glue. Again, it's the nice thing about this paper, this water-based glue is that it's very forgiving to use. And yeah, here we go. The next one's finished. Again, this is a little bit of a, how do you say, yeah, a nice afternoon pastime when it's raining. You can, yeah, very relaxing activity. Yeah, and so, but let me read a couple of questions here again. Yeah. I was just thinking yesterday about doing the same thing, but instead of using paper to cover everything, spray painting the entire slide, but masking the center, front, and back. That's interesting. And never, honestly, that's actually quite an interesting one as well. What you could do is maybe you use this kind of a hole punch, and then you kind of maybe put some kind of a tape, a round tape, over here, and then kind of spray paint the whole thing. Why not? Some kind of airbrushing, maybe. Yeah, that would be interesting. Yeah. After all, this whole thing is a hobby, and there is it's not just hardcore science. It's also art, of course, and looking at the exploring nature and looking at the things that you see under the microscope, and so doing a little bit of this creative, yeah, tinkering around. It's of course something that it's also part of the hobby. Yeah. I actually have a couple of compressed cardboard slides made in a similar way to those wood ones, but at least structurally speaking, they have a removable cover slip, so you can manipulate the sample too. Cardboard slide, that's interesting. Never heard of those. Okay, yeah, interesting. Yeah. So yeah, so I got 2 of them done. Yeah, maybe I'll make another 1 or 2. I was thinking of a suction cup for the mask. Oh, okay. So no chance of anything seeping under. I found some nice pink glass spray paint on Amazon, pretty cheap. Honestly, that's interesting. Yeah. So what you do is you kind of cover this whole thing up, and then you kind of spray over it, and then you can also give it some kind of a nice, yeah. Please try it out. Send me the picture or whatever. I'll share it online. Yeah? Honestly. Yeah. Yeah. So that's second one here that we've got. Maybe I'll make another one just for the fun of it, and I've got 2 more, and then I'm gonna show you some of these here, and then I'm going to move on. I'm gonna show you some of the other things that I've 3D printed, just in case. I don't know if you know what this here is. You will never guess, but this is something that I 3D printed, which I'm gonna show you later. Okay, it's actually something for the microtome. Yeah, that I've got. Yeah, and it kind of works. Yeah. Okay. If it works, I will update you via email for sure. Thank you. Thank you. Okay. Please. And if you allow me then to share this, it would be really nice. Yeah. I have so many projects on the go at once. Yes. I'm kind of happy now because, in case you don't know, I'm of course in my real non-microscopy life, I'm a teacher. This means the summer holidays are now starting in a few days. So it's still very, very busy because as we're finishing off the school year, but then it's going to become a little bit easier for me. And then this is when I'm going to do again a couple of more microscopy projects. So what else? Yeah, we're going to just try it again. Here we go. Again, yeah, this might be actually indeed a thing. Maybe a spray-on PVA glue. Would be indeed a possibility here. So let's do it like this. Yeah, okay. We have to remove this again here carefully. Here we go. Yeah. So that's not pollen. Press down a little bit to make good contact. Okay. Just for you, because I mentioned PVA, very often those white wood glues are also PVA based. right? I don't know about this one specifically, but the ones that I have over here is I used also water-based glue as a mounting medium. So here we go again. Yep. So next one, maybe try it like this. Like this. That is actually also a possibility. See, again, I should have actually cut out this part here first. Dang, again, I forgot. Yeah, doesn't matter. This is just a proof of concept a little bit. Yeah, so I don't know, maybe you're just gonna do it like this. Yeah. Not too much. And here, just like this. So here we go. Or, I don't know, use a sharp knife to cut it out. Okay, I should have, see, this is a failed project, a failed slide. Yeah, so what I might want to do now is I'm not going to put the other thing right over it. I might actually just let it dry like this, and then I'm going to try to cut it out carefully using a sharp knife. And then I'm going to scratch off carefully the glue that's on the glass, and then I'm going to add this one here again. But I think you get the idea right now, okay? So that's basically what I've done, and just very quickly. And the next thing that one might want to consider is maybe instead of just using plain blue paper or whatever color you want, maybe to actually, maybe not, why not print a pattern using an inkjet or laser printer or whatever. Plenty of things to experiment around here, okay? Yeah. Okay, there is, okay, just somebody, Scott, just joined in. He was just reading the comments, okay? So that's basically what I just wanted to show you very quickly, okay? You know what, just for the sake of how do you say, of rounding everything off, I just wanted to show you how they look like under the microscope. And then I'm going to show you quickly some of my other projects as well. So that's basically here, the onion one. Let's quickly have a look, because of course every now and then we also want to observe directly something under the microscope. Let me see, does this work? Oh, it's slowly coming. Switch on the microscope, how's that? Here we go. It's a little bit off center, I just realized. Okay. So, yeah, it's basically as expected. Yeah, that's the brown onion skin, and what you might be able to see here, those tiny little squares that you see, these are crystals. Yeah, those tiny squares in here, right? The ones over here and here, these seem to be so-called oxalate crystals. I've already made videos about that, right? But that's basically, just so that we also see under the microscope. Oh, here it's actually nicer to see them. So it's just that we see under the microscope, how it actually looks like. Time check. 1:28 UTC plus 8 hours. So it means I'm in the middle of the night. So just for those of you joined in, it's basically just today. I wanted because I did not have time yesterday. I just wanted to not miss another week, and for this reason I moved it to today, but at a slightly different time. Okay, so what I would like to do now is I would like to show you. We can later on, I've got some water samples here as well, but I would like to show you what this actually is here. Okay, and I'm going to make this a little bit bigger. There will be a separate video on this. But these are actually specimen holders for plant stems, okay? And that is something that I 3D printed for my microtome, okay? And you see a little bit that there are basically different cutouts, and I'm going to demonstrate, because then it's going to be easiest for you, right? So let's, this here is my microtome, okay? You probably have already seen it before. It can be clamped on the table. And the problem now is how in the world am I supposed to put a specimen in here, right? And the official instructions are, what you do is you basically take a carrot, the vegetable, and you put the carrot in here and use the carrot as a specimen holder. So if you want to cut something, a leaf or something very thinly, you have a little slit in the carrot, and basically the carrot will hold the specimen. And this kind of works, right? But it's pretty messy. And so what I've been thinking about is how can I have some kind of a specimen holder for plant stems, which some of you might have already seen before. I made some videos as well. So these are some plant stems in alcohol. And what I've done here is I've actually 3D printed those holders here. And those holders essentially have a different, on the 1, 2, 3, 4 different cutouts for plant stems. And you see that those they touch the side here. So basically this one goes in here, right? Like this, and then here on the side I have the stem, and then I'm able to use a sharp knife, and that's another new thing that just arrived recently. This is a shaving knife used by barbers, but with an exchangeable blade. In the past I used the other razor blades, but they were kind of too flexible, so I bought myself this type of shaving that they are really sharp. And then what I can do, I'm going to demonstrate this also. I'm able to kind of cut it off, like this, right? And those 2 holes in the middle, look, I've made those here in order to kind of be able to pull it out again. And so I've made different holders here, essentially for different diameters of stems. Now you might wonder, no, why did I put them on the side here? Why not in the center? And the reason is because I want to use the side of the microtome here as a support. So you're just going to see in a second when I show you. So essentially then when I cut it like this, then the side of the microtome, the edge over here kind of gives it additional support. So this was a little bit the strategy here. Okay, and it kind of works. Okay, it's not perfect, right? But I just want to demonstrate this as well, and then I can again show you a couple of cross sections under the microscope. And then I have also again an aquarium sample here, and I've got a moss sample where I did not find any tardigrades yet. So these are basically the things here. So the only problem that I have is when I want to cut this, it's very, how do you say, unstable, right? Because normally I clamp it on the table like this. This is the table. But that's a little bit the problem. The top face should be printed against the bed. This one over here. Well, the thing is, if I guess I don't know if what exactly you with this. The thing is if this is too high, then I'll be cutting somehow the plastic. So that's a little bit the question here. So I just want to demonstrate this. So what I've got here is that's these are some flower stems that I've got, and I'm just going to pull out one of them here. It doesn't really matter which one. All right, and I'm just going to use this here. This is in alcohol, so this means that unfortunately it's also going to dry very quickly. And now I have to find one that kind of fits fairly tightly. So maybe this one here is fairly tightly. And look what else am I doing. I actually made a little hole here, and here I've got a pin. And I'm going to push the pin all the way through to kind of hold the stem like this so that it's not able to move out. So, but I think I made the hole a little bit too small. Okay. So, see. Uh-oh. I just realized that I'm not able to push it all the way through. Okay. So here we go. Okay. So basically, I'm not able to pull it out anymore because the pin kind of is fixing it, all the way through here. And then what I do is I just push it in here like this, right? All the way to the bottom. Okay. And I want to cut it like this. So then you see that the edge of the microtome is kind of stabilizing it as well. And this is now going to be a little bit unstable because it's not fixed on the table, right? Careful not to stab yourself. That's indeed the case. But I did, cut off the tip of the needle, just to be. Otherwise, it would have been too long. But it's indeed. So, but now I have to be careful that I don't cut myself, right? Because this is actually a really sharp razor, and again, the angle has to be correct. And what I've really why I wanted to use this is because of this metal edge that I have here, which kind of makes it inflexible. Oh, I just want to show you something else that I made. For regular razor blades, I also made a guide like this. Yeah, so these are the regular razor blades, and I also 3D printed this here, right? So it's also possible to kind of maybe use this as to cut, but I like this one a little bit more because it's longer, yeah, and therefore it gives me a little bit more flexibility, because if I move across too much like this, then this is not stable anymore, and then I do not get a good cut. Okay. So I'm reading comments. I didn't even think that we are sitting proud of the printed holder, so smoothness doesn't matter. Okay. So let's yeah, that's okay, yeah, because this one is kind of lower, yeah, lower down. So let's give it a try. And what I'm doing is I actually might want to also give it a small cut on the other side here first, yeah, simply so that it's easier to cut. And then I just do the following: I kind of slice it across like this, okay, and then I turn it up a little bit. 1, 2, 3, 4, 5, 6 notches. 1, 2, 3, 4, 5, 6 notches, and I just realized I need a little bit of water here. And then sometimes I'm able to catch it and sometimes not. 1, 2, 3, 4, 5, 6. This is a little bit of how do you say a little bit unstable, I have to admit. And then as the alcohol evaporates, this is, I don't know, for whatever reason, this is not very good to cut, but I suppose it's because to do with the stability. Okay. But I was able to get fairly repetitive thin cuts like this. And I can imagine maybe because also it's not sufficiently wet. Yeah, and it's shrinking a little bit as the alcohol evaporates. Okay, this one was a more decent one. And then you have to drop it into a little bit of water. Okay. And I'm just going to put it directly on a microscope slide. Let me quickly add a little bit of water first. Because as the alcohol evaporates, it will shrink significantly. Okay. Okay. Nah, this is not a... Yeah. You have to carefully shave it off a little bit, but okay. I can also try the following over here. Okay, this is also a possibility. The advantage of this here is, it has an advantage and a disadvantage. The advantage of this here is that the blade is more visible. So this means that the specimen, as you cut it, is able to slide on top of the blade. And over here, it's not quite as large. Yeah, so it kind of gets a little bit jammed up. Yeah. So, but this one over here, I can put on into the water again. You can see actually because of the changing surface tension. And let's try this one here again. Yeah, see. So sometimes a wet specimen is insofar better because it doesn't quite shrink as because water does not evaporate quite as quickly as alcohol. Okay. Again, this is just a short demonstration, and then if you basically, you see it's kind of moving up, and then you can pull it out again, yeah, and that's basically how large it is now. Yeah. So this is again not a, I would say, what I would sometimes also recommend is maybe take this and just drop it into the water for a couple of minutes or an hour or so for the alcohol to be replaced. And then when it's wet again, then you can also try to cut those. I'm just going to put this under the microscope again, just to show you again. This is a little bit just a very quick demo So I don't know how good this specific specimen is. And I need a cover glass. Where did I put them? Here they are. Cover glass goes on top. Yeah. And I'm just going to put it under the microscope and we're going to have a look. And it's another, where is the scope? Wait until it appears. Here we go. And this is how it looks like. Okay. Those dark dots that you see are air bubbles. Yeah. Let's go up with the magnification a bit. Yeah. But generally, I have to tell you, I'm fairly happy with the result. Maybe some of you remember a couple of weeks ago I also made several of these cuts, but just today I wanted to show you just a little bit this tiny little improvement that I made. Huh? Yeah, so I made a video about staining those a couple of weeks ago, but when you actually focus through it, you can actually see that it's not thicker than a cell layer. Yeah, so I only see one cell layer, which actually shows that it's fairly thin. Yeah. Yeah, so there's a comment again, polarization live view. A wave plate can be made from the clear CD cover. Oh, interesting. Better color with polarized light. Rotate the top, lowest polarizer, rotate. Okay, so that means a clear CD cover. I was actually thinking about ordering a wave plate, a lambda force plate. Yeah. And there's another question is, do you have a video on how to prepare the stems from fresh, preserved in alcohol? What I have done is actually very easy. The thing is the following. These are stems from a you might want to switch back here. These are the stems from a non-woody plant. So basically from a flower, it's called the sword lily. Who cares? It's just a flower. The stems originally were quite green because they were photosynthesizing chlorophyll, was so very dark in that sense. And what I did is I simply cut the stem and dropped it into more or less concentrated alcohol, ethyl alcohol. Right, a very cheap one, I left it in there for a day or two, and then everything turned very green because the alcohol removed the chlorophyll, the green pigment. I dumped out the alcohol, added fresh alcohol, and I kept on doing this 2, 3, 4 times, and then that's basically how it looks like now. Okay, and you're able to keep it in alcohol for many years. This one over here, for example, this is one of the first one I kept. I'm not kidding. I made this over 20 years ago. Yeah, and I kept it in this bottle now for over 20 years. So this was actually from a tulip flower. It's actually one of my first trials of preserving the specimens. Same thing, and you can see, however, that it decolorized much more than this one over here. And it stores in the alcohol indefinitely, really, right? Again, but the difficulty or the problem with storing it in alcohol is that it evaporates very quickly, and as soon as you take it out, it will start to shrink quite a bit because the alcohol evaporates and it will become quite hard, right? So for this reason, I sometimes maybe recommend that you actually drop it in maybe some water, and then it will absorb the water, and then it might be easier to cut. Yeah. So the live stream time slot is now slightly more convenient. Formerly, I have to set an alarm. Now I just have to stay awake late. Okay, yes. Well, the thing is, normally I do the live streams on a Saturday. Saturday, quite more or less in the evening, because that's usually the time when I'm also back home. And just because there's been a request for different, different time, and that's why. And because I did not have time yesterday, that's why I tried it today. Simply. But I'll see whether this is more convenient or the other one. I don't know. Yeah. So yeah, I'm pulling out the needle again, the pin. Yeah. It's a little bit crooked. That's why it's also a little bit difficult. Yeah. And this one over here is again the stem, and yeah, it has already shrunk quite a bit, and it's becoming harder and harder as it starts to shrink. Yeah. But this one is not too short, so I'm just going to throw it away probably. Yeah? So that's basically a little bit the thing that I just wanted to show you. What else do I have here? I've got here remember this one over here from before. Let's have a look at the pollen as well. I need to change the how do you call this, the scope view. Okay. So yeah, that's basically the pollen. Yeah, in nail polish, not the best, how do you say, mounting medium. The problem with nail polish is that you can see there are lots of bubbles, right? But this is again one of those experimental slides. Yeah, it looks kind of nice with the polarized light and DIC. That's why you get all those rainbow colors. Yeah, yeah. Yeah, but that's essentially how the slide looks like. So I'm willing to spend under, wow, $2,300 on the used inverted fluorescence microscope. Okay, do you think that it would be a good idea, or can advise me where do I find offers? Oh, gee, this is such a difficult question, honestly. What's a good idea? It depends what you want to do with it, and if it's a hobby, you know, if microscopy is a hobby, and if you want to do this as a pastime and as for recreation, essentially, yeah, then you don't have then you have to do that what interests you, right? And what gives you the most enjoyment. So I cannot answer it if it's a good idea or not. It depends how you define your hobby, right? I tell you the following: fluorescent microscopy is interesting, but I mean, you're working with UV light. You have to have, if you really want to exploit it properly, you have to use proper UV stains and so on. So I don't know to what extent you want to actually, how deep you want to dive into the whole thing. That's a little bit more the question that I have. But this is not something that I can give you a recommendation or recommend against it. Generally, as a how do you say, as a general rule of thumb, as we always says, I generally recommend. Especially if I don't know how experienced you are, but for beginners or for not so experienced people, just to stick to the standard equipment until you've gained experience, and then once you actually know what you want, then you can always specialize and buy different microscopes. That's kind of my general philosophy, but I'm not saying that this is valid for everyone. Yeah. So this is really, I get a lot of questions about what microscopes do I recommend, and I tell you that's very difficult. It's because a lot is personal preference, and as I always say, the best microscope is the one that you end up using the most often, right? But your question is where can you get those? eBay is indeed a possible. Oh, it's already recommended here, eBay or Surplus. Yeah. But again, you see, you don't know what you're getting sometimes. Yeah. Yeah, so that's a little bit the thing. Yeah. Just reading again here. Yep. Okay. Yeah. So that's basically just quickly. I'm just also occasionally have a look at the time, how far I'm actually going, and let's desk view. So yeah. For those of you just joined in, a very quick summary of what I've been doing. I've been paper covering some of the slides right at the beginning. Then I looked at the microtome here, right, with a little bit of showing you some of the things that I've been experimenting around with. Yeah, and I would like to now move on. I would like to also show you a couple of other samples, yeah, because sometimes people are not only interested in the technology aspect and specimen prep aspect, but also a little bit in just observing some samples, even if we don't see a lot. You always have to close this. It's very dangerous. Yeah. By the way, those blades, I think I mentioned, can be exchanged. So there are spare blades as well that you can buy, and they're pretty sharp, right? So that's also something that I've been playing around with. So let's put this away. And I do have two samples here to look at again. Okay. Sample number one. This is basically, I've been using my Spearman funnel again to extract some living things out of moss with limited success. Just want to show it to you, and I want to tell you also a little bit about the background of this. I did make a video about the Spearman funnel already before. Okay, but for those of you who have not seen this, just want to show it to you. I've got some moss in here covered with water. Now most of the water has been absorbed, and basically there's a sieve in here, and then down here you can see that water bears, nematodes, other microorganisms and soil particles, whatever, they fall down, right? And there's a clamp here, and then basically it can be collected. And you see over here, this is kind of the moss water. that I kind of collected, and I'm just going to show it to you, but don't be disappointed, okay? And I will tell you why I think the result is a bit disappointing. The reason is because the moss was quite dry, but yeah. So unfortunately, I've not been able to find water bears, moss piglets, but I'm just going to show you in any case. So if you want to take up a sample here, you do the following. You take a pipette, you kind of cut it off here to make it a little bit larger, so it's easier to take up also some slightly larger particles. Okay, and here we go. Okay, maybe a little bit, slightly more. And usually what I do, in the first trial, I just put it under the microscope as it is, even without a cover glass, simply to get an overview, because there's a lot of material now, yeah, in the center here. I'm not going to do that now because it's too, otherwise there's the danger of me making my objective wet. I drop a cover glass on top here, yeah, and this kind of spreads a little bit the soil particles and whatever, and we're going to have a look at this, right? Yeah. Again, this is not a very active sample, but yeah, I have to make careful that I don't misclick here. Okay, I have to wait until for the camera to get to come on. For whatever reason, it takes a little bit longer. So here we go, right? And that's how it looks like. Yeah, so it's the sample is 1 day old, and the moss was very dry, right? And normally what I do is I kind of scan through the whole thing and try to find some living things here, right? And occasionally we actually see that some of them were actually able to survive the dryness and they came back to life. This is still at a fairly low magnification using my 10x, 10 times objective, but it was actually, yeah, not a very, yeah, not a very active sample. Yeah, I expected more worms, for example. I didn't find any water bears. I did not find, but there were occasional ciliates visible here, as you see right now. So this is a little bit the thing. And if you want to, I have not done this yet, but normally what I usually do is, in this case, you make a time-lapse video and you speed it up. And when you do that, then sometimes you're able to see the movement much more, and then you're actually able to realize that, yeah, even in a sample like this, there's actually more movement going on than what you might want to expect. Yeah, yeah. But that's a little bit, I would say, the thing that where you have to be a little bit patient, and then if you kind of scan the sample, then occasionally you're able to find some more or less interesting things, maybe some amoeba, occasionally some water bears, not in this sample. And one of the reasons why I think that this was not so successful here is because I collected this sample from from moss that was dry. We'll tell you a little bit later on exactly from where I've got this. Just a second. Do you see this here? This here is, for example, looks very much like a so-called conidia. These are basically the spores of a fungus. Let me show you. The arrow up here, this one over here, it looks typical for conidium. Usually fungi look like this. Yeah. In the background, sometimes you see some tiny dots moving around. Sometimes there are bacteria present. So what I would like to normally do in this case is I'm now going to switch over to phase contrast, because phase contrast makes bacteria better visible. Okay, I have to, yeah. And this is basically now the thing where we see bacteria. Now I would like to also answer one question that I received just either by email just recently. And because actually you see, wow, how many bacteria there are, right? Which we did not see before. And the question that I got was the following. Somebody was doing some soil microscopy and was not seeing any microorganisms. He says, how is this possible? And are there none there? And the short answer is chances of soil generally is highly active, right? But you're just not able to see it with a microscope. Why not? Because the bacteria are generally too small, or they are of too low contrast. And what I've got now is phase contrast, and now we're able to see them in different shapes, right? So you see there is a lot of life here. but maybe not quite easily visible. I'm just going to show you now how would this look like in bright field. I'm going to also close the condenser. Okay. Yeah, you still see that there's a little bit of movement, but the contrast is by far not as good, right? So you need to have a pretty decent microscope already to be able to see those bacteria. But the best contrast is, of course, achieved with phase contrast, and that is then a different type of technique that you're using, right? So basically just wanted to this way also answer this question is why don't I see any microorganisms? And I say, well, probably they're there, but probably they're just mainly bacteria. If they're not moving, then they're also more difficult to see because sometimes things are more easy to see, of course, when they're moving around, right? Obviously. So let me go to a couple of comments here. New topic. I would love to see a session on methods for visualizing cilia and flagella. Oh my gosh, I think you're talking about silver staining, because this silver staining method is actually a way of actually visualizing them. Counting and measuring often is needed for identification of many eukaryotic microbes. Correct. Yeah, as a matter of fact, Mr. Feusner, he's late Professor Feusner from the University of Salzburg in Austria. He was also one of those protozoologists who was involved in so-called silver staining of ciliates. This is a staining method where essentially you're able to make the cilia visible. The cilia, the tiny little hair on the surface of a cell, and by observing the patterns of these cilia, you can do some basic identification. Right. Nowadays, I would say the gold standard of identifying microorganisms these days, of course, is DNA sequencing. But of course, that's not available to us. I have to tell you, I have not done any silver staining myself. That would be, again, something that, maybe over the summer holidays, would be a possibility. You have to use silver nitrate, and then basically by treating them, by staining it with silver nitrate and doing a chemical reaction, and somehow metallic silver will be deposited there where you have those cilia, and then you can actually see apparently quite nicely the different rows of cilia, and depending on the patterns, you can actually do some identification. Mr. Feusner is late. Professor Feusner, he was actually quite much involved in this type of staining. Yeah. Do you have any advice on how to harvest pollen for microscopy? Yes, I have done this already. Basically, there are different ways how you can do that. The easiest way is simply to collect the answers of a plant and then simply dip the answers on a microscope slide, and you've got plenty of pollen. You can also try to extract pollen from honey, but then you need a centrifuge to do that. I've also done that. Several months ago, you have to check this channel here. There are actually some videos where I did that. Yeah. Okay. Yeah. Jesse is mentioning, tap the flower on the slide or collect using a cotton bud. Yeah. I would like to put some dog blood under the microscope. It doesn't look like human blood. Actually, it's the blood of a mammal. It should look very similar. Yeah. So I wouldn't know why the blood of a dog would look different than human blood. Okay. You lead vapor. Okay. Lead vapor for staining. Yeah. How do you set up Köhler illumination? Okay, that's actually another thing here. It's a little difficult for me to explain now because I don't have a camera system here. And actually, in most cases, the cameras are the microscopes are already set up. But if you kind of want to know, let me just show you. I'm now using my low power microscope. Low power. How do you say? Let me do the following. Just a second. Let me set up everything. Maybe I can actually show you. Look what happens when I change the Köhler diaphragm. It's a field diaphragm. I'm able to, I can actually focus this here as well. Here we go. Now it's properly set up. So that is basically what I'm doing right now. Is not the condenser aperture diaphragm, but it's the so-called the field diaphragm or Köhler diaphragm. And this one is actually used to limit stray light. So what you do is basically you just open it enough that the whole field of view is illuminated. Yeah. and then basically you get the maximum contrast. Yeah? So Köhler illumination was actually invented back in the day, 19th century, at the time when essentially the illumination system was not as even yet. So there was a problem. They had this filament, and then you could actually see that the filament of the lamp was actually visible sometimes in the image, right? So this didn't look very nice. So August Köhler, he basically invented the system to even out the illumination. And nowadays we still have it because it gives you a slight edge in contrast because it reduces stray light. Yeah, so that's a little bit the thing here. So, but in most cases there is not a lot to set up really. What you have to do is you have to center it. It's not right now because the video it's centered in my microscope, but it's not centered here on the video. But there are actually two screws. Look, what I can do is I can actually move the adjust the center position of the condenser. Yeah, and then you have to focus. That's by raising and lowering the condenser. You have to make sure that the edge of the Köhler diaphragm is in focus, and then you have to open it up, yeah, and then it's set up, yeah. That's pretty much it, huh? Yeah. So so it's explained here as well, yeah. So. Yeah, centering is also something that you normally want to do. As a matter of fact, in some microscopes, if you don't have Köhler, it really kind of looks washed out, the image, right? So that's a little bit the thing that I just wanted to show you here. So that is, and you now want to know a little bit where I collected this moss from. And video is in the making, but I collected it from a tree. From which tree did I collect it from? An oak tree. Here it is. Okay, don't worry, I did not rip it off from the tree. Don't do any damage to the tree. I'm going to show you the desk view, the square desk. Okay, so that's an oak tree. I found this on the ground. And why? What's so special about this tree? Well, last week when when I did not do the live stream, I visited with my family. Styria, which is a federal state in Upper Austria, and over there, right out somewhere in the middle of nowhere, there is a tree, and it's the oldest oak tree in Europe. That's what they claim it to be. It's 1,000 years old. It looks like a pretty nice, decent, big tree. And essentially, it's the second time that I'm visiting this tree. Of course, there's a sign there, and there's some kind of a resting place for you to have lunch there, and so on. And a very nice looking tree. And on the ground, this is what I found, right? Because I don't want to damage the tree. So, and on the tree, I also had there was a little bit of moss growing that I also collected. Yeah, and so this is basically from apparently from Europe's oldest tree. Wait for the video. Video is in the making. The only reason why I did not make the video yet is because, then that's the next thing that I want to show you, I had problems cutting this hard oak wood, because oak wood is very hard, right? So in order to soften it, I tried the following. Look, and I don't know if it works, but I took the oak, small oak branch, and I put it into glycerin. So it's this oily looking substance, and apparently if you leave it in here a couple of days, then it will soften the wood, because apparently the glycerin will infiltrate the wood and break open the hydrogen bonds, and makes the wood softer for cutting. I don't know if this works, okay? So it's kind of experimental as well, but I just wanted to show you a little bit of what is also in kind of going on in my microscopy lab. And that's also now you know also the reason why I made those things here, because I would like to cut the wood this way. Now there are also other protocols that mention that you should cut the wood and then you should boil it, because boiling it also makes it softer. Did not try that yet, but that's also something that I would like to try. But right now I'm just trying the glycerol part. Yeah, it's not the only one. I do have a couple of I do have a couple of branches here. Yeah, so that is basically from the I've got plenty of sample material. Look, there are also some lichens on here. Yeah. Again, I did not damage the tree. Don't worry. I got this from the ground. Yeah. How often are you discarding and replacing or starting fresh the stored water samples? I suppose you're asking Basically, when I collect pond water in a jar, how often am I replacing it? Well, I'll be honest with you that some of them I've been keeping now for several months, and there's always water evaporating, and so I add a little bit of, keep on adding a little bit of more tap water to it. So essentially, yeah, months maybe. Yeah. And it depends a little bit on how stable the jar is. Yeah. So that's I'm just having a look again here. Okay, yeah. So that's basically another thing that I wanted to show you. Okay. My best samples with giant purple amoeba are now almost a year old. Oh, that's cool. Yeah, yeah. But again, the how do you say, the moss water? Unfortunately, and the reason I think is because I had the best success in finding tardigrades with moss that is constantly somewhat moist or wet. Moss that is exposed to dryness. I've generally almost consistently been not successful finding tardigrades. So if you don't find tardigrades very soon in a moss sample, chances are pretty good that it's a bad sample. Go out, collect some fresh moss from the shade somewhere which did not dry out, and chances are pretty good that you're able to find more. Yeah? Would tap water harm the water sample since it might have chlorine in it? Definitely, yes. So the tap water that we have got here that I've got here is not chlorinated. But what is sometimes also recommended is if you don't, if everything's chlorinated, either you let it stand for a couple of days for the chlorine to kind of escape. However, there might be certain heavy metals in the water because of the plumbing and the pipes and so on. And for this reason, I sometimes also recommend. Oh, thank you. And for this reason, I sometimes also recommend that you use, how do you say, just bottled water without carbon dioxide. Yeah, and you don't want to exchange the water too much, but always add a little bit of water. Yeah. Yeah, interesting to see the progression of biodiversity. Yes. So what I have over here also, I made a video just recently talking about the aquarium because there is an aquarium, not in my home, but in the school where I work, which was full overgrown of algae. And my colleague who was taking care of the aquarium, he said he exchanged a little bit the substrate of the aquarium, and then all of a sudden he had this algal bloom, and I collected some of the water plants and algae, and this was a few days ago already, and so maybe we're gonna see a little bit more now. Usually what I do is I don't keep those samples very long, maybe just for a couple of days, and then I observe again a little bit, make another video every now and then, right? And then, if I don't see anything, I just basically, this is very, very primitive what I'm doing right now. Okay, this was very primitive. And then I usually dry wipe it. Just a second here. I add a cover glass. Maybe I'm gonna add a larger cover glass. And usually this is probably a bad sample in any case because I don't see a lot of algae because most of them will be actually growing on the surface of the plants. But usually after a couple of days, you have again some kind of a progression, and sometimes you're lucky and you're also able to see something of interest. Okay. But this one is probably not going to be a very good one. Let me see, and it takes again some time for the camera to switch on. Okay. Yeah, so there is some capillary action still going on as it's spreading. You know what, I'm going to add this here. I'm going to put this here. I'm going to go up with a little bit of light. Yeah, and what we're also seeing quite a bit is, a whole bunch of bacterial growth. Yeah, there are, a couple of, this is a rotifer, of course. Might want to have to, but again, I would say, a whole bunch of diatom shells. Yeah. And that's basically also something that I occasionally like to do. I just take a sample and then simply, as I mentioned already before, just observe the progression of microorganisms. Occasionally you make some interesting discoveries. Yeah. Yeah, but as you can see a little bit, the density is not quite high, and that's of course because it simply tapped the plant material and it kind of rinsed out whatever was on there. Yeah. I don't recommend mineral water because the minerals will precipitate out over time and can kill samples. Okay, well, I guess, yeah. I mean, I guess it depends a little bit on which type of minerals are in, because if you've got, I don't know if this is still a thing, but what I heard is that if you have still old metal pipes somewhere, then it might be the problem that those metals from the metal piping might actually also kill microorganisms. Yeah? So that could be also an issue, right? So if what you can try in any case is if you've used tap water, then let it stand for some time, for at least for the chlorine to escape. Yeah? Yeah. So that's kind of the thing I wanted to show you here. What I would probably do here is the following. Yeah, before I'm gonna actually maybe I might try that. I'm going to do the following. How am I going to do that? I'm going to show you. You know what? I'm going to show you. I think I'm going to try that. Let me see if I find a tube. Is this? I'm going to find this tube over here. This seems to be clean. Let me just a second. Just a second. Yeah. Yes, I've got it. This is. I might need to buy a little bit more. Dangerous stuff. Let's try something dangerous today. Okay. What I would like to do, and this maybe I'm able to report back by next week or so. What I'm going to do is I want to isolate all. This is going to be really brutal now. What I'm going to be doing. Okay. I want to isolate all of the diatom shells that are on here. And I'm going to do that using hydrogen peroxide. 30%. This is aggressive stuff, okay? We got to be careful here. So, but before I dump it out, I'm just going to give it a try. And I'm going to show you now what I'm going to do. I'm going to take this here as it is. Diatoms are growing on here, a lot of them. And I'm going to put it into this tube here. And this is going to be now extremely cruel and brutal what I'm going to do. Okay. This water, yeah, I'm going to discard. And now, and this is going to take a couple of days maybe. So maybe I'm going to report back next week. I got to be really careful. %30 hydrogen peroxide. I'm going to put a lid on top because I do not want to accidentally spill it because it's so aggressive. And what's going to happen now is the following this hydrogen peroxide is going to oxidize pretty much everything away over the next couple of days. And the diatom shells, which are made of silica, they will fall down, accumulate on the bottom, and then I've got a nice sample of diatom shells. Okay. I should have gloves and eye protection on. You're probably correct. Now, what's going to happen when I get this on the skin? I've already got it on the skin a couple of times. The skin is going to turn completely white. It's going to just basically burn away the skin. You do not want to get this stuff into your eyes. Okay. So what's going to happen is slowly we're also able to see that there are now bubbles appearing. That's oxygen. Because the hydrogen peroxide will also break down. It's going to oxidize pretty much everything, and yeah, the diatoms are going to fall down on the bottom and will be collected. So that's something that I will report back on next week. Okay, so I'm just going to put it on here, and this one here, I'm going to carefully put it away. This is 30%. 3% is used for disinfecting. So many hand disinfectants contain actually hydrogen peroxide, but at a much, much lower concentration. Yeah, yeah. So got to be careful with this. Okay. So just saying. Yeah, that's why it's also not so easy to buy those things. They don't want to sell it to you. But can you use 3% hydrogen peroxide? Yes, also possible, but you have to be much more patient. Yeah. Yes. When I open this here, I am aware of this. when I open this here, because of the oxygen, there might be pressure in your and it's going to flush. Yeah, that means you have to be a little bit careful here. Yeah. So just saying, and now you can also see slowly. I don't know if you're able to see this. There are some more and more oxygen bubbles appearing. Yeah? So, but that's something that I just wanted to also show you, and yeah, gloves and eye protection. That's something I have. I don't want to mention this. You shouldn't be doing it, but I've had. You got to be super careful. You got to be super careful with. I've already had concentrated sulfuric acid on my skin. In the chemistry lab, it burns away, your skin turns yellow. In my lab coat, if you kind of have these droplets of acid and all of a sudden you've got holes in your lab coat, it looks like completely normal at the beginning, but then when you wash your lab coat, all of a sudden your lab coat starts to fall apart, because where the acids broke it away. So indeed, you got to be sometimes a little careful with the stuff that you're working with, right? So what I'm going to do now is the following. I'm going to... do I ever wear gloves? Essentially, when I do microscopy work, actually not. Okay, because I generally say if I do not want to work with infectious material, right? Which is actually dangerous. I mean, if I've got, for example, moss water and things like this, I mean, this is stuff that I'm normally exposed to anyway when I essentially am in the garden and so on. But what I do, where you have to be really careful is if you start to enrich microorganisms by adding food, for example, then you have really explosions of microorganisms. And then I do observe basic hygienics. Yeah. In the lab, I remember when I worked, we did the radioactive labeling, and also I worked with highly carcinogenic, cancer-causing substances, especially when doing DNA staining. And sometimes we had to wear double gloves, in case one of them breaks, because it's so, dangerous, right? So, so this is something I just wanted to mention. Yeah, I got a couple of questions here again. So I'm going to try to, maybe I answered most of them already. I'm just going to go through this here. Yeah, so that's basically what do you use as a, how do you say, support for punching this here? Leather could be, what I found out, it should not be too soft, otherwise it's going to warp the whole thing. What main factors affect the microscope resolution? Honestly, if you want to be a little bit provocative about this, then I would say the sample or the specimen itself. Very often that is underestimated. Essentially, if the specimen is not good, then also the quality of the image is not good. So I would say that sometimes, and that's a psychological issue as well, sometimes the resolution is low, but it looks good because the contrast is high. Yeah? So this is a little bit also an interesting thing that I would like to mention, that it's sometimes microscopes with a good resolution essentially, might look not so good because the contrast is too low. Yeah. So what kind of microscope is indeed complex? Okay. Bankrupt credit card also works for. Yes, I use this cardboard for spreading the glue, right? But you can of course also use a credit card, and I think in the past, a couple of years ago, I even used a microscope slide. For spreading for spreading it. Also works. Yeah. Yeah. So. I was just thinking yesterday about doing the same thing, but instead of using a paper to cover everything, spray painting the entire side. Yeah, instead of spreading it, you can also use some kind of spray paint. I tried different types of glues as well. I kind of like the paper-based glue, than the water-based glue. Glue stick, you have to really... It didn't work quite as well, I think, because you have to use a lot of this glue from a glue stick. Yeah, a couple of compressed cardboard slides. I talked about that already. Suction cup, basically spray painting it by covering this with a small suction cup and then spray painting over it. Okay. I'll watch your polarization live view. A wave plate can be made from a clear CD cover. I did not try that yet. Okay, but I know that plastic like CD covers are indeed also polarizing. Slightly more convenient, so the timing is better for some of you. Ethanol, I always use ethanol. It's cheaper, but if you have isopropanol available, I think the reason why sometimes ethanol is also better for these things, at least as a first step, is because ethanol and water mixes fairly well. As a matter of fact, if you want to make it completely free of water, you should not, as a final step, you should actually take it out from the ethanol and put it into isopropanol, because isopropanol is not does not attract water as much. And for this reason, by actually using isopropanol, you can remove even the remaining water. This is actually used before embedding it in the paraffin. It has to be completely free of water, and then you use, as a last step, you use isopropanol, but it's more expensive. Yeah, so I have it, but I'm not using it. Yeah. So that's already mentioned about this. Should you use a fluorescent microscope? And I said, well, difficult for me to answer. Generally, I think whatever gives you happiness and satisfaction is, yeah. But I mean, $2,300 is quite a large amount. And I would say that maybe the biggest problem of buying used microscopes is you don't always maybe know what you're getting, right? So that's a little bit sometimes the issue, right? Yeah. Okay. For visualizing cilia, I talked about this, the silver staining method. Yeah, polarized filters. Okay, yes. Oh, cool. Yeah, I'm still. By the way, I did. I also several weeks ago bought some polarizing filters to do some 3D imaging. I'm still working on that. Okay, I didn't forget about that. But it's possible to also get a stereoscopic view using a regular microscope using polarized filters. But I'm still working on that, and I don't know exactly yet how I'm going to show this on screen, as actual 3D view. Yeah. Do you have any advice on how to harvest pollen? You just take a little bit of pollen and tap it on the microscope slide. It's really that simple. Yeah. Okay. I would like to put some dog blood. I don't know if I don't know if dog blood actually would look so different under the microscope. Yeah. Curly illumination also quickly showed to you. How often are you discarding and replacing? Yeah. I mean, I've had some water samples that I had quite a long time, and then I just wanted to throw it out. So usually what I've done is I just observe it the last time under the microscope and then I dump it out, because we've got several ponds and lakes in the area, so I have easy access to many more water samples. Yeah, so that's the thing. Yeah, I just want to have a quick look at some other comments here. My understanding was isopropanol is hygroscopic and over time pulls water from the air to 65%. Interesting. I mean, I thought that ethanol will stabilize at about 96%, and I heard somewhere that that isopropanol contains less water than ethanol, and that's the reason why you usually use isopropanol as a final step. But I could be wrong. Okay, so I read somewhere that isopropanol apparently is more drier, but maybe before it started to pull water. So that would be kind of interesting. Yeah. You can do a shareable 3D on screen using red-blue system. I get it. Yep. Yeah. So that's something with the red-blue kind of how do you say, glasses. Yeah. It contains less to start, but it does seem to pull water from the air more. Okay. Yep. 30%, so 70%. Okay. Yeah. Yeah. So ethanol dries your samples more though. Okay. Yeah. What I'm generally not how do you say measuring out anything. I'm just dropping it in and just exchanging it. So I'm generally not doing anything quantitatively, right? So what I'm going to do now is I've been talking nearly 1.5 hours already. A very quick summary for those of you who joined in a little bit late. Just to show you what I've done is I just wanted to demonstrate to you how to make very quickly. So it's a little bit sloppy. Those paper covered slides, more a proof of concept. However, there are a couple of links in the description, which actually goes a little into more detail. That's number one. Number two, I showed you the 3D printed thing that I made, for this microtome here. So I made a specimen holder for stems, yeah, for plant stems, yeah, of different sizes, which can go in here because originally you're supposed to use some kind of a carrot or whatever, right? Yeah, and then I also have done a little bit of microscopy. I wanted to show you some of the Biermann funnel and some of the things that I kind of yeah, extracted from here. Was not able to find any tardigrade, no water bears, unfortunately. Yeah. Then last but not least, the most dangerous thing here, I'm trying to extract. Ooh, look, now it's going. Isn't this beautiful? Do you see all of those oxygen bubbles here, right? Now it's really oxidizing. I quickly let the pressure off. Okay, there wasn't a lot of pressure. Yeah. Okay, there's a question here. Paper covered? Yeah, there are indeed. Back in the day, 100 years ago, it was quite common. And I've got a few of those if you joined in late. These are antique paper covered slides. Okay, maybe over 100 years old. They look quite nice. Yeah, and this was fairly common at that time. And I simply very quickly simply also tried to do those. Yeah, this doesn't look quite as nice as the others here, right? But I just wanted to show this to you because it adds stability, also for artistic reasons, for collectibles, right? And for branding purposes. Yeah, and of course it does make the slides a little bit more stable as well when dropped. Yeah. So that's just yeah, for yeah. And if you're more interested in paper covering, please do read the article that I linked below, which I found, because it goes quite a bit in more details, yeah, on how paper covering can be done, right? And if you're interested in trying this yourself, I did include a PDF and a PowerPoint file also in the link. Yeah. So I'm just reading again some of the comments here. I hope I didn't forget anything. Otherwise, I'm going to do the following. Otherwise, I'm just going to slowly check here what am I... okay. OCD. I have OCD and I always wear gloves. Yep. Generally, I would say the following. That's a little bit my philosophy. I don't want to play around or mess around with things that I don't fully trust, right? So I would never, for example, use sewage water, right? Because there's really a lot of bacteria in there. I also would not, for example, rot some meat. Yeah, and put those bacteria under the microscope because that's probably not so good. Yeah, because bacteria that are able to grow on meat maybe could maybe be more infectious, right? I don't mind so much putting fungi mold from plants under the microscope, right? So you see, that's not a hard rule or so, but generally I do try to be a little bit careful and I try to avoid things. I don't mind putting pond water samples under the microscope because we just generally swim in ponds anyway, right? But that's kind of a little bit this approach that I try to do. I do not do so-called hay infusions, even though this is a very common one. Also historically what they do is people take hay, which is dried grass, and then basically you can grow yeah, also paramecia and other microbes this way. I don't want to do that because the bacterial concentration is too high. So yeah, and it's not necessary. But yeah, so I'm just saying, yeah. Okay, yeah, I would. I think what I'm just gonna do now is I'm just going to call it a day again. Yeah, I hope that was interesting for you. Please do leave your comments. I'm still a little bit. How do you say? Yeah, struggling a little bit with this YouTube channel. I mean, the live streams I'm going to keep, no question. But in the last couple of months, the views have dropped significantly. Could also be because it's summer and people kind of like to go out and watch less YouTube, which is actually quite nice. But that's a little bit one of the things that I've been thinking about is which direction or which videos to make. And if you have any suggestions concerning that, I would be very happy as well. But the views have really dropped down, yeah, on some of the videos. Yeah, but I'm going to call it a day. I wish you all the best. Yeah, enjoy microscopy. Happy microbe hunting as always, and hope to see you again next week. And if for whatever reason it still doesn't work out, I always have this how do you say this this timer, not timer, but this kind of this trailer as it's called, which will basically tell you when the live stream will start. Okay. Okay, I'm going to call it a day now. And yeah, I'll report back next week on how this actually worked out. Okay, bye-bye, and happy microbe hunting, everyone. All the best.